Good morning, everyone. You can hear me?
Good morning, Dr. Bar. Yeah, we can hear you.
Great. And let's share the PowerPoint. I'll share you my plan. Can you see the PowerPoint now?
Yes.
Okay, we were doing beta oxidation. And we were here. Let's do that. I'll start at 10. Great. So do you have a class? What is your next class today? You have any class today?
We had anatomy supplemental from 9 to 10, and then we have physio supplemental.
When?
Um, I think 1 to 2 and 2 to 3.
Okay, great. So we have good time. So you had a supplemental till 10. So I'll give you 5 minutes, okay? We'll start 10:05. Is that okay?
Thank you.
Oh, what's the beeping sound? Okay, I hope you get some noise-free learning. Diego? I think because I'm more ready today. So I'm like all tied up, ready and tidy. I think the topic is complicated. That's why I highly recommend today's recap, especially because I'm not going to rush like yesterday. I always feel bad holding students for long. So I'll share my plan, but I'm just giving time for everyone to join because you had um, a supplemental for anatomy. I'm going to share my plan. I'm not going to go fast with this section. It's very complicated, but I'll make it easy. Only thing is I'm not rushing today, please. I'm not rushing with this session. Yes, we had a session yesterday. I posted a announcement on Canvas. It was 5:15 to 6. Please look out on your Canvas. If I'm holding any sessions, I do tell your class reps, but see, your class reps are doing their best. Sometimes you might miss it, but I also post second announcement on Canvas page. So look at both places, okay? I will tell you at the end of session, which is then when will be the next session. I'll start at 10:05, okay? I'll just give everyone time to join. These slides have been shared with your class rep for extra help in the drive. This is known as CP2 lecture content recaps. This is more about what topics students might struggle with. No, there are different slides. These slides are known as CP2 lecture recaps. I hope it's available on the drive. I shared it yesterday with
Is
your
it
class.
different? Is it different than the one under CP2 exam review slide?
Yes, because that's a whole review, which is a team review. But this is my extra help. Because what happens is on Canvas, what review is posted is for all three campuses, right? But they are not taught like I teach line diagrams. So that's why my line diagrams are in a separate recaps. Does that make sense? I don't want to
Yes.
overwhelm other campus students with my line diagrams because they have not gone over it, right? Before the exam. Uh, this is shared. Is your class rep in the room? Zoom room? Any class rep in the Zoom room? Teresa, Rahul?
Yeah, Dr. Blas, I'm here. I actually don't have an email from you with those slides uploaded. Could you resend it?
Oh, what about Teresa? But I sent it yesterday.
Yeah, I know. I just don't have the email. So maybe if you resend it, I can upload it really quickly.
Okay, what about Teresa? Just a second, guys. Give me a minute. I didn't know this. Okay, I shared yesterday. Okay. Anyone has it? So Teresa might have shared it. No? Let me resend it, please. I wish you would have told me earlier, but anyways, let's do that now. I'll forward you again, please, right now. Rahul. Please let me know you have it so you share it with all right now. So because I want students to have it before I start it, please.
Yeah, thank you. Do you know
No
what the title of
worries.
the slide presentation would be?
Sorry,
What would you title the slide presentation?
I'm just sending you right now.
Okay, thank you.
Right now, I just resend you. Just let me know if you get it. I re-forwarded you. Can you please share with them so then I will start? I shared it yesterday after the session. Everybody is good, so let's start and not waste time. Did you get it, Rahul? You have it.
So is it titled CP2 Lectures Recap?
Yes, yes,
Okay.
yes.
I'm gonna send a link to everyone in this chat. They can just access
Please,
it through there.
please. I'll give you a minute, guys. Okay. Please send it to everyone. All good? Shall we start? Everybody is good? Yes, everyone is good? You want me to start? Everybody has the PowerPoint now?
We have the link now. You can start. Thank you.
Everybody is good. So please see what is my plan. First of all, I'm not rushing today. Somebody said it's a complicated topic. Absolutely, it's a complicated topic, but I'm simplifying it. This session will be 1 hour, 30 minutes. If you want a break after 1 hour, I will give you, but I'm not going to rush over this content, please. Please see that what was my plan is to get off major metabolism off your plate. Please do that for me this weekend. Finish fatty acid synthesis, fatty acid oxidation. Both of them also include tag synthesis and tag lipolysis. Finish lipoprotein and cholesterol. If you are done with these 4 lectures this weekend, you are good for CP2. Don't worry about phospholipids and icosanoid at all. I'm going to come in the interactive session and review it. Please do not miss any interactive session next week because that would be your second review. So let's see what did I do. I did fatty acid synthesis. Always compare synthesis with oxidation, fatty acid synthesis with oxidation, tag synthesis with tag lipolysis. So tell me beta oxidation or fatty acid oxidation. Now some students are confused. Are you ready? Everybody is ready, right? Unable to open file. Can anyone else open it? Are you okay with the file? So who is on Arturo account? Excuse me, what's going on?
I was able to open it. It's working fine for me.
Oh, your question was for Amir. So I'm sorry. I sent this email yesterday night, right after the session. Please, it's not me. So you got people got get organized. Your class reps got to send the file as soon as they get it. Okay, can I start the session, please? This was sent last night. Thank you, please. Okay, so let's see that. Okay, let me get in my Zen. You got to understand that one thing that I struggle with, I'm very organized. I do things organized way. I want everyone to please work with me in an organized way so everybody has things at the right time. Please understand, okay? Now let's go to the interactive session. Let's go to the supplement. Let's get in your zen, okay? So what I was telling you is you compare fatty acid synthesis with oxidation and you compare tag synthesis with tag lipolysis. What you got to do? So let's do beta oxidation. First of all, when you are doing fatty acid oxidation, you need fatty acids. So please do not forget to turn on the tap HSL. If you will not get fatty acid, how will you oxidize it? You're not going to bake sale to get fatty acids. So you're dependent on one enzyme that will give you fatty acid. So what is that enzyme which will give you fatty acid in a low insulin state? What is that one enzyme that will, I'm glad, Amir, you got it. What is that enzyme that will give you fatty acid in a low insulin state? Give me the enzyme HSL. Very good. So you got to first turn on the HSL tab. What do you want to do for me in these four metabolism? Keep the right enzyme in the right pathway, the right state in the right pathway. Is it well fed? High insulin. Is it low insulin? So who will tell me what's low? What does low insulin mean to you? What do you mean by low insulin? When are you in low insulin state? Any state? Fasting? Fasting is later. Before that, no, not everyone fast all the time, right? First it is low insulin status in between meals. Yeah, during sleep. I hope you're awake today. In between meals, during sleep, fasting, starvation, diabetes. You got to get familiar with the low insulin state. Whenever you talk about low insulin state, please don't stop there. You got to understand which antagonist hormone is high in this low insulin state, because not all hormone works at the same time and are not primary. Please don't overload your brain with information. Rate limiting enzyme, primary hormone, primary substrate, primary product. Then you go over the pathway. There's no point knowing in this specially section steps that are not clinically relevant or physiologically relevant. It's not about glycolysis. You draw the whole pathway, pin it down on the paper and feel good. No, cholesterol is not about that. No more steps which are physiologically linked, clinically linked, linked to drugs, linked to rate limiting enzymes. So low insulin you should get familiar with. When I'm saying low insulin, I'm also asking you to know antagonist hormones. So tell me HSL, which are the antagonist hormone of against insulin that activate HSL? Which hormones do you talk in activating HSL? It is a low insulin enzyme, but epinephrine, very good. I like that. That should be top on the list. And second in the list should be cortisol. So you also got to delineate what are the primary hormones if it is not insulin. Insulin means well-fed state. So beta oxidation is definitely a low insulin state. Don't forget to turn on the HSL, which will give you fatty acid. Now, fatty acid. Fatty acid in the circulation is different from tags high in the circulation. Also, please know why students get confused is they think they think in well-fed state, if fatty acid synthesis is on, fatty acid oxidation is shut down. That's not true. Both pathways are active, except in well-fed state, one is more active than the other. None of the pathways shut down in your body, please. So that is why, yes, in a low insulin state, beta oxidation or fatty acid oxidation will be more active than fatty acid. In low insulin state, fatty acid oxidation will be more active than fatty acid. What? Can you fill in? Synthesis. Also, please know, don't get confused. Beta oxidation and long chain, since this is the major oxidation, that's why when they say fatty acid oxidation, they talk about beta only and long chain only. Understand that. So yes, you have to turn on the fatty acid tab. So tell me, if you have too much fatty acid in the circulation, which enzyme is active? If there is too much fatty acid in the circulation, which enzyme is active? Yes, very right. Somebody said that one pathway is active, other is less active, but none of the pathways is shut down, except when there is an imbalance. Then the other pathway will be more and more active depending on the state. So tell me, if fatty acids are high in the circulation, which enzyme is active? Which enzyme is active? No, see, you have to relate them. HSL, right? Do you mean HSL, Diego? Yes, HSL, hormone sensitive lipase, absolutely. That maybe was a little scary, but yes, it is HSL. So when HSL is active, then there's too much fatty acid in the circulation. So when do you expect HSL to be active? When there will be too much fatty acid in the circulation, any state, any pathological state, and any pathological state that HSL will be active. Any pathological. So give me a pathological state. Not fatty liver, please. Don't go wrong. Fatty liver is alcoholics. No, no, no. So I'm saying HSL is active in a low insulin state. Yes. So give me one pathological condition where HSL will be diabetes. Absolutely. Primary first. In diabetes, because there is an insulin defect, there HSL will be highly active. That's why today what I'm going to do is first know normal to know abnormal. So you put on the HSL tab normally in a low insulin state. I will do diabetes too. I will do fat metabolism defects too. But then once fatty acid is released, what is it picked up by? Which two main tissues? Which two main tissue it's picked up by when fatty acid is released? Liver as well as not adipose. Mainly muscle, liver and muscle. Let's do primary first. That doesn't mean I'm not talking about adipose. Now look at this. I'm telling you adipose is releasing fatty acid. Am I right in a low insulin state? Is adipose releasing fatty acid in a low insulin state? Yes or no? Is adipose tissue releasing fatty acid? And when I'm asking you who is picking that fatty acid and someone is asking adipose, why would adipose release fatty acid and pick it back again? Is there any purpose? Adipose should release fatty acid and for what? For other tissue to use. So when I'm saying adipose tissue in a low insulin state is releasing fatty acid, it has to be picked by tissues other than adipose. So your answer adipose cannot be correct. It is liver and muscle. Once they get into the liver and muscle, since it's a low insulin state, fatty acid will undergo oxidation. Don't memorize every step, know the phases. First step is activation. How do you activate fatty acid? How do you activate a fatty acid? By adding a CoA. Exactly. But this CoA is impermeable to inner mitochondrial membrane. So what do you need next to push this fatty acid into the mitochondrial matrix? No, citrate is for fatty acid synthesis. Here we are talking fatty acid oxidation. I'm not talking about fatty acid synthesis. Carnitine, absolutely. You need carnitine shuttle. What are the components of carnitine shuttle? Carnitine as a carrier, CPT outer membrane, CP2 intermembrane. There is also one more carnitine acyl translocase, but it's not physiologically or clinically significant. That's why we mainly talk about CPT1 outer membrane, CP2 inner membrane, and carnitine, because these three are physiologically and clinically related. What do they do? Push fatty acid inside the mitochondria without a CoA. Then CoA is added and the fatty acid will undergo oxidation. Now, why are fatty acid undergoing oxidation? Because please remember, I am telling you, don't do two states at a time. Don't do two pathways at a time, but do compare and contrast. Fatty acid synthesis, oxidation. TAC synthesis, lipolysis. So when you are in a low insulin state in the liver, what will fatty acid undergo? Oxidation or synthesis? Or TAC synthesis, oxidation. And what is the major enzyme? Now, don't forget the rate limiting enzyme. So which enzyme of carnitine shuttle is rate limiting and is a gateway for control of beta oxidation? CPT1. But what is the major enzyme? So remember, there's a rate limiting enzyme and there's a regulated enzyme. Which is the rate limiting enzyme? So citrate. Somebody answered citrate. Please don't talk about citrate in fatty acid oxidation. When you compartmentalize just like these pathways, you get good grades. All you know what to do is, this is my pathway, these are my enzymes, these are essential components, and this is the state. Don't flip-flop them, right? Citrate, you talk in fatty acid synthesis. So this is fatty acid oxidation. So what should be the major enzyme, tell me, that turns on oxidation? What is the major enzyme in your body that does oxidation? There's an enzyme class. No, there's a class of enzyme that always does oxidation, and its subclass, dehydrogenases. They are the major enzymes that do nutrient oxidation. So what is the enzyme here? Fatty acyl-CoA dehydrogenase. Please, this is not acetyl-CoA. Students are confused. Acetyl-CoA is what? What is acetyl-CoA? How many carbon is acetyl-CoA? As 2 carbon, it is part of the TCA cycle, starting molecule for many syntheses. But this is acyl-CoA, which means activated fatty acid is known as acyl-CoA. There are different acyl-CoA. There's a short and medium chain acyl-CoA. There's a medium chain acyl-CoA. There's a long chain acyl-CoA. That's why you have a case on MCAD deficiency, which is medium chain acyl-CoA. There are different dehydrogenases for different chain fatty acid. One more I was telling you is what is rate limiting and regulated. So tell me the rate limiting enzyme for fatty acid synthesis. Rate limiting enzyme for fatty acid synthesis. ACC. Is there a regulated enzyme there? Is there a regulated enzyme there? Which one? Which is regulated? FAS. Similarly, what's rate limiting in beta oxidation? CPT1. But there is one more enzyme which is also key, which is acyl-CoA dehydrogenase. Very, very important enzyme. You know why? Who will inhibit acyl-CoA dehydrogenase? Tell me. What levels will inhibit acyl-CoA dehydrogenase? What levels will inhibit acyl-CoA dehydrogenase? High levels of? No, there are levels. Dehydrogenases are always inhibited by high levels of ATP or not hormone. I'm talking about intermediates. NADH, very good, absolutely wonderful. All dehydrogenases are inhibited by high levels of NADH. Do you know where this happens? In alcoholics. That's why alcoholics drink and sleep. They can use any fuel for energy. Alcoholics also accumulate high NADH to NAD, and they inhibit this enzyme, acyl-CoA dehydrogenase. They can use fat, right? But what are the products? Whether you burn carb or you burn fat, what do you get in the end? Whether you burn carb or fat, what do you get in the end? In the end, you get a lot of, in the end, you get a lot of what? What are the products? What are the products of carb and fat burning? You get a lot of, anyone? Acetyl-CoA, ATP and acetyl-CoA. So you get acetyl-CoA and ATP from burning carb as well as fat, but carb, you get it in a well-fed state. Fat, you get it from in low insulin state. So whether you burn carb or fat, you get a lot of acetyl-CoA and ATP. You don't have to memorize the number, but fat are concentrated fuel. So are you aware of all these enzymes? Should you keep them in fatty acid synthesis or oxidation, all in pink? Should you keep them in fatty acid synthesis or oxidation? They are all for oxidation. And remember, how do you start? Always you start fat in a low insulin state by turning on HSL. Now you must be, I think I want to correct that. I asked you thioesterase and thiolase. Remember yesterday? Thioesterase is where? Fatty acid synthesis or oxidation? Thioesterase, fatty acid synthesis. Thiolase is fatty acid oxidation. I want to correct that. It's not ketone. Thiolase is fatty acid oxidation. All it's doing is, please remember, just as a fatty acid is synthesized in 7 steps by adding 2 carbon at a time, it's also broken in the same 7 steps by removing 2 carbon at a time. And who is removing the 2 carbon? Thiolase. Who is removing the two carbon every step? Thiolase, right? This we did, right? So when you are low energy status, do you want to do fatty acid oxidation or not? Tell me. When you are in a low energy status, do you want to burn fat? Do you want to burn fat? Yes. Why? What will that give you? What will that give you? Burning fat will give you a lot of ATP is a concentrated fuel. So that is why, please know, just as I taught you fructose 2,6 bisphosphate for gluconeogenesis, glycolysis, I taught you, I'm teaching you now in fats, citrate and malonyl CoA. Please, these three intermediates are important because they make the cycle so that they don't become futile. Can fatty acid synthesis and oxidation go on at the same time? Can fatty acid synthesis and oxidation go on at the same time? No, it's futile exercise. So what arrangement body has made? If you have enough of malonyl CoA in cytosol, what will that favor? If there's a lot of acetyl malonyl CoA in the cytosol, what will that favor? Which pathway? Which pathway will it favor? Lot of malonyl CoA fatty acid synthesis. That's why malonyl CoA, high malonyl CoA in the cytosol turns off or slows down which pathway? High malonyl CoA in the cytosol slows down which pathway? Slows down. That's why I'm not saying shutting down. Do you understand? That's why I never use the word shutting down. So high malonyl CoA in the cytosol slows down which pathway? Fatty acid oxidation. And which is the gatekeeper there? Which enzyme does malonyl CoA inhibit? It inhibits fatty acid oxidation, but which entry enzyme it is inhibiting? CPT1. So you got to be careful about these intermediates. They have a specific site. Yes, malonyl CoA controls such that you are not doing fatty acid and oxidation at the same time. Yes, none of the pathways shut down. One is active over the other, or the other one is slower than the other. Secondly, this slide also teaches you, can you do genesis without ATP? Can you do any genesis without ATP? No. And look at this. In a high AMP state, which enzyme is inactive? That's why in high AMP state, which enzyme is inactive? What is this enzyme for? When they test you on the enzyme related to the pathway. So in a high AMP state, which enzyme is inactive? What is ACC an enzyme for? Fatty acid synthesis. You cannot do genesis in a low insulin state. Does that make sense? Let's stay positive, right? Let's stay positive in life, or we can move on. ACC is the enzyme for when they test you on the enzyme related to the pathway. ACC, rate limiting enzyme for fatty acid synthesis. So will insulin turn it on and citrate turn it on? Yes. Are they the positive players? Tell me one which is missing here. ACC should be turned on by insulin and citrate. What else? Remember, ACC is active when polymerized. So ACC will be biotin. I like that answer. I'm glad you don't forget your vitamin. Biotin is required. Insulin turns on. Citrate, not CO2. ATP is an excellent answer. Insulin, citrate, ATP, and biotin. You can't forget these four for fatty acid synthesis. So let's stay positive. And okay, ACC, sure. So citrate, insulin, ATP, and biotin, ACC activators are essential for ACC. We have learned the role of malonyl CoA. Now let's do the disorders. There are a lot of disorder Dr. Panini has explained, but I'm going to simplify it. Just a second, I have too many windows open. So let's do this. Let's do diabetes. In diabetes, which will HSL tap be on big time? In diabetes mellitus, will HSL tap be on big time? Will it be turned on? Yes. And when it will be turned on, where is it turned on more, in type 1 or type 2? Type 1. Because type 1 is absolute insulin deficiency. That means this is on much higher in type 1 than type 2. It doesn't mean type 2 does not have HSL active, but it's just in type 1 HSL is too much active because there's absolute insulin deficiency. But don't forget that these fatty acids, when picked up by liver and muscle, is there any difference? In liver, normally fatty acid burning supports which two pathways? In liver, fatty acid burning is prerequisite for which two pathways? In liver, fatty acid burning is supportive of which two pathways? Ketogenesis and gluconeogenesis. Absolutely right. So if HSL tap is on, what will increase in circulation of a diabetic patient? If HSL tap is on, glucose and ketone bodies. That's why please know that diabetics suffer from, especially type 1, hyperglycemic and hyperketotic coma. Now let's look at the fat disorders. This is about diabetes mellitus. Let's do fat disorder. Now tell me, look at this. If acyl-CoA dehydrogenase is inhibited, will glucose and ketone go down in circulation? It's suppose this is when, yes, very right, very right. When the prerequisite for ketogenesis and gluconeogenesis to be on is that the fat should burn in the liver. Also, tags are not only mobilized from adipose, could be mobilized from liver as well as muscle in a low insulin state. So tell me, if you have acyl-CoA dehydrogenase deficiency, will glucose and ketones go low in your circulation, in the circulation of that individual? If acyl-CoA dehydrogenase is defective or deficient, will glucose and ketones be low in circulation? Yes, that is why you have a case. Now, acyl-CoA dehydrogenase is many types. It's long chain, medium chain, short chain. Also, please know, which I want to tell you very clearly, that only long chain fatty acid need carnitine for transport. Short and medium chain do not. How is this clinically relevant? If there is a carnitine defect, what can you give in a diet of such patients? Only long chain fatty acid needs carnitine. So if somebody has a carnitine defect, what can you give in a diet of such patient? Which fatty acid will you give so that they are not deficient? Only long chain needs. Only carnitine, long chain. Yes, you will give short and medium chain fatty acid in the diet of individual who has a carnitine defect because they cannot do long chain fatty acid utilization. Short and medium chain fatty acid do not need carnitine for transport. Other disorders I will address. Okay, so now let's see, if you have acyl-CoA dehydrogenase deficiency, what will be low in circulation? Glucose and ketones. Where do you see that? MCAT deficiency. But understand, only fat metabolism disorder that affect liver will have hypoglycemia and hypoketones. Will fat metabolism that affect muscle have low ketones and low glucose? You have to know that is fat matter is this. So you got to delineate disorders, but understand, is this disorder primarily affecting liver or muscle? So if a fat metabolism affects liver, will glucose and ketones be low in circulation? First of all, if fat metabolism defect of, yes, but if fat metabolism defect affects muscle, will glucose and ketone be low in circulation? No, what will be low? Only and only ATP. That is why there are different signs and symptoms of different disorder that Dr. Panini has enumerated, because some disorders primarily affect liver, some disorders primarily affect muscle. So let's understand, tell me, if there is a disorder, fat metabolism disorder primarily affecting liver, what will you see? What would be the sign and symptom? If there is a primary fat disorder, if there's a fat disorder primarily affecting liver, what would be the signs and symptoms? Hypoglycemia and low ketones. Yes, but if there's a if the fat disorder primarily affects muscle, what would be the sign and symptom? Decreased exercise tolerance, myoglobinuria. But to which exercise? Aerobic or anaerobic? Aerobic or anaerobic? Muscle fatigue, but to which exercise? Aerobic. Do you want to differentiate this from McArdle's? Yes. McArdle's also have decreased exercise tolerance. They also have myoglobinuria. Why there's myoglobinuria? When you have, when the fat metabolism, or the whenever your muscle is affected, why there's myoglobinuria? Whenever muscle metabolism is affected, why there is myoglobinuria? Because muscles are acting against insufficient ATP. There is muscle cell lysis and there is myoglobin in urine. So muscle fatigue, decreased exercise tolerance, myoglobinurias are mainly due to metabolism affecting muscles. But in case of fat defect, the decreased tolerance should be to aerobic exercise. If it was GSD, it would have been decreased tolerance to anaerobic exercise. Which GSD affects muscle? Which GSD affects muscle? Do you recall? GSD affecting muscles? McArdles. So you got to compare and contrast later on boards. They compare and contrast the muscle metabolism defect, whether it's related to carb or fat. So now let's do the disorder. You understood the difference between the two disorder. Now Dr. Panini has enumerated disorders. So let's see first of all which disorder affect liver. MCAT deficiency, liver. CPT1 deficiency, liver. Jamaican vomiting sickness, akki fruit. They have a poison known as hypoglycemia. They also affect fat metabolism in liver. So will you see hypoglycemia in all these three disorders? Will you see hypoglycemia and low ketones in all these three disorders? That is why the first presentation of MCAT deficiency is hypoglycemic hypoketones, low glucose, low ketones, right? What else? One more thing is there, please, whether you have okay, I'll do that with carnitine. Let's first do acyl-CoA dehydrogenase deficiency. Hypo. When fat metabolism affects liver, there is hypoglycemia and hypoketones. That's why MCAT deficiency, you will see hypoglycemic, hypoketotic coma. Diabetes, you see hyperglycemic, hyperketotic coma. But look at this. There is a fat metabolism defect that affects muscle, which is which one? CP2 deficiency. CP2 deficiency affects muscle. So in CP2 deficiency, what will you see? Tell me. Give me the signs and symptoms. Since you know normal, now tell me abnormal. In CPT2 deficiency, what would you see? Presentation. There will be low ATP is not a presentation, right? You can't see ATP in a patient. Muscle fatigue, myoglobinuria, decreased exercise. Myoglobinuria is spelled as MYOGLOBINURIA. It's not urea. It's not urea creatinine, please. Myoglobinuria, it's not urea with it, right? I'm just correcting someone has in the chat. So what will you see in CP2 deficiency? You will see muscle fatigue, decreased exercise tolerance to aerobic or anaerobic exercise. Aerobic or anaerobic exercise. Aerobic exercise, CPK raised, CK enzymes is also raised. But tell me what is conclusive? This is an enzyme deficiency. Okay, there are signs and symptoms. What is conclusive? What will deposit in the muscle? Glycogen or tags? Glycogen or neutral fat? Tags, that is conclusive. That all gives you it's a fat metabolism defect. Please know tags have a different stain. Also, tags are the most neutral lipid. All the polar portion of glycerol is masked by fatty acid. Most polar is tag. And now the last thing, in MCAT deficiency is about which oxidation? Tell me. Which is the most common oxidation? MCAT is about what? Did I tell you there's alpha, beta, omega, and peroxisome? Which is for very long? Which is for very long? Very long is for which one? Which organelles do very long? Because you have to cut down very long into long. No, very long needs what first? Peroxisomes. Peroxisome. Beta and long chain most common. MCAD is also beta, please. Alpha was for branch chain phytanic acid. Alpha needs additional hydroxylase. What was the disease that Dr. Panini has connected with phytanic acid alpha? Infantile or adult refsum? Infantile or adult refsum? There's a chart. What is for phytanic acid, which is alpha, branch chain fatty acid? Adult refsum's disease. Very good. Adult refsum's disease. And then there is omega. Let's see what is omega. Omega is last carbon, right? So MCAD is first of all beta oxidation deficiency. Whenever beta is also normally, please, omega is for short chain, but omega is for last carbon. Pathologically, omega oxidation is increased when there is no beta oxidation. So when I was asking you in MCAD deficiency, what is defective? It's beta oxidation that is defective. When there is no beta going on to compensate, just like thalassemias, other globin chain rises, other oxidation rises. Look at this. If omega is oxidized, will you have 2-carboxylic acid? So whenever there is no major oxidation, omega turns on. In omega oxidation, will you have 2-carboxylic acid? In omega, will you have two carboxylic? That's why they are known as dicarboxylic acid. So when you see MCAT deficiency, you will see hypoglycemia, low ketones, dicarboxylic acid. MCAT deficiency is also predisposed by sickness, illness. Not just like that, there's a predisposing factor that they become symptomatic. And also they suffer from metabolic acidosis because of dicarboxylic acids. So you need to understand, when is omega turned on? No, omega is for short chain. Omega oxidation, the oxidation is turned on when beta oxidation is missing in the body. When beta oxidation is defective, beta is for long chain. Yes, beta, long chain. See, no. I'm not saying beta is for long chain. Somebody has a question. So there are the fatty acids are classified by chain. So there's long chain, very long chain, short and medium. Very long, more than 20. Long, more than 12. Short and medium, less than 12. But oxidation is more common of long chain. Long chain needs carnitine. Omega is for short chain. When I'm saying beta oxidation, in the long chain is more common. Okay, does that make sense? The long chain more common, and in that beta carbon is more common. Yes. Yes. So when beta is not happening, omega turns on. Now, don't do the whole thing, but please understand carnitine. First of all, carnitine, shuttle has carnitine as a carrier, CPT1 outer, CPT2 inner. I will differentiate these three. So when carnitine is not working, what will accumulate in the body? How will you see that in circulation? Because carnitines are not working, what will be high? Acyl carnitines will be high, medium chain, long chain. But remember, in one deficiency, carnitines, acyl carnitines cannot be high. Primary or secondary carnitine deficiency? Primary or secondary, where acyl carnitines cannot be high? Primary or secondary? Primary or secondary? Primary, because there's no carnitine, right? First of all, secondary carnitine deficiency because of secondary causes, where vegan diet, because carnitine sources meat and meat products. CPT1 liver, I have told you, different signs and symptoms. CPT2 muscle. MCAD, I already told you, low glucose, low ketone, dicarboxylic acid, accumulation of medium chain acyl carnitines. Acyl carnitines will accumulate everywhere in fat metabolism defect except primary carnitine deficiency. Now, don't do all of them, but remember that all of them are related to peroxisome, Zellweger, infantile, or X-linked. So when there is so when they are all related to peroxisome, which fatty acid you cannot use? Whether it's Zellweger, infantile, or X-linked, because they are all related to peroxisome. You don't need to memorize biogenesis assembly, but they are all related to peroxisome defect. So what you cannot utilize, tell me, which chain fatty acids you cannot utilize? If peroxisomes is defective, you can use. If there's no peroxisomes, very long chain, which is more than very long chain is more than 20. Adult refsums, I told you, alpha defect. Adult refsum is alpha defect. Branch chain fatty acid, they need hydroxylase, right? Please know, I will redo all this. I just want to bring your attention. The cell is important, though fat oxidation is mitochondrial. So who cannot use fat as energy? Because this is very important compartmentalization. Fatty acid synthesis, cytosolic. Fatty acid oxidation, mitochondrial. So tell me who cannot use, and very long peroxisomes. So tell me who cannot use fatty acid as fuel. More than 20 is very long, yes. Brain, only long chain, because long chain is selectively permeable to CSF. What else? Who cannot use fat as well as ketone as fuel for energy? RBCs. RBCs lack mitochondria. Not muscles. No, muscle is not the right answer. Brain, only long chain. RBC, brain cannot use long chain. RBC cannot use it at all because they lack mitochondria. RBC are entirely dependent on glucose. They can neither use fatty acid nor ketone. Please note that's why compartmentalization is important. Brain can use ketone bodies, absolutely, 100%, because that's an alternate fuel. What's the primary fuel for brain? That's an alternate fuel. Glucose, yes. Why we are saying brain cannot use fat? Because remember, most common oxidation is beta and long cannot be, because of selective barrier, cannot be used by brain. So brain, glucose and ketones, RBC only glucose. That is why normal is important. Normal is important to know physiology and alter pathology. Now, even chain and odd chain, don't keep drawing this. My diagrams are to help you understand. If you break even chain fatty acid or odd chain fatty acid, in the end, the product will be 3 carbon. If you break even chain fatty acid or odd chain fatty acid, the end product will be 3 carbon compound or intermediate. When you break even chain or odd chain fatty acid, which is known as propenyl CoA. Lot of vitamin here, so please no propenyl CoA. Vomit. Anything, vomit, vomit, vomit. VOMIT. Anything can converge into this pathway. What is the clinical significance of this pathway? First, keep the right enzyme and vitamin in the same box, right? So first is carboxylase. Do all carboxylase require biotin? Which other carboxylase you learned requires biotin in fat metabolism? Which other carboxylase? ACC. ACC is an enzyme for which metabolism? When the fatty acid synthesis. Do propenyl CoA carboxylase also require biotin? But this is not for fatty acid synthesis. What is this for? What is this in fat for? Even or odd chain? This is what we are talking, even or odd chain fatty acid oxidation. Odd chain fatty acid oxidation. So tell me in biotin, though vitamin deficiency, please know that when you do this fat metabolism lectures, stick to the enzymes which are clinically or physiologically relevant. Don't draw the whole pathway. It will not make sense. Rate limiting enzyme physiologically, clinically with drugs, pay more attention on that. So tell me, though vitamin deficiencies are not severe, carboxylase or isomerase deficiency, which will reduce methylmalonyl CoA in body? Carboxylase or isomerase deficiency, which will reduce methylmalonyl CoA in body? Carboxylase. Know the enzyme deficiency disorder steps 2. When there is enzyme deficiency, what accumulates? Substrate. What goes down? Product. So please understand that. So don't mix match the enzyme, don't mix match the vitamin. So biotin deficiency will lead to reduced methylmalonyl CoA, not B12. So we will do vitamins in spring, but understand these 2 steps separately, right? It's in carboxylase or biotin deficiency. What will happen to methylmalonyl CoA? High or low? Carboxylase or biotin defect. What will happen to methylmalonyl CoA? High or low? Low, right? And also odd chain fatty acid can form glucose, not even chain. Even chain fatty acid do not form glucose. Odd chain fatty acid can be glucogenic. We're gonna common fatty acid broken down. Okay, I don't understand. You mean types? So beta oxidation long, you're talking about the types? Yes, quickly. Beta oxidation, long chain fatty acid, beta oxidation, long chain, omega oxidation, short chain, alpha oxidation, branch chain, peroxisome, very long chain, peroxisome, very long, alpha short, beta long. Okay, sorry, let me redo that. Beta long, alpha, branch chain, omega short, and peroxisome very long. Did you get that? Yes? Omega is short and alpha is branch, which is phytanic acid. Ketones. Liver synthesize ketone bodies but never utilize them. Please know that. Ketogenesis, primary site, liver. Its mitochondrial, can RBC use them? No. What else can use ketone bodies? RBC cannot. Brain uses them 100%. What else can use ketone bodies? RBC cannot. Can anything else that cannot use ketone bodies? Liver. Liver synthesis. No, not muscles. Liver. Liver forms ketone bodies to be used by extrahepatic tissue, which is brain and muscle. But liver and RBC, please note tissue specifications and a little why. Liver cannot use ketone bodies. They synthesize ketone body, but they cannot use it because of this enzyme, which is absent in the liver, which is actually used for adding CoA. That is why you need this enzyme to add a CoA missing in the liver. So RBC and liver cannot, mature RBC and liver cannot use ketone bodies. Now, ketone metabolism is well fed or low insulin? Ketone metabolism is well fed or low insulin? Ketone metabolism. Well fed or low insulin? It's a low insulin pathway. Very good. The same thing. Low insulin pathway, you turn on this big tap, HSL, you release fatty acid and glycerol in the liver, fat burning support ketogenesis. Ketone, who's the starting molecule? What's the starting molecule? What's the starting molecule for ketones? Acetyl-CoA. Now tell me, when I'm saying ketone metabolism in a low insulin state, give me the states where ketone metabolism will be high. So first physiological, then alter physiological, then pathological. Please go this way in metabolism. Don't jump to MCAD, diabetes. You got to understand first the pathway, compartmentalization, well-fed or not. Yes, yeah, I'll give you, let me finish this, I'll give you break. Let me finish this. But can you tell me first what are the low insulin state for ketone? Just give me the states: sleep, in between meals, fasting, starvation, and diabetes. Great. I'll give you a 5-minute break. Is 5 minutes good? Or you need 10 minutes? How many minutes? 10, okay. Okay, is everyone ready now? Everybody's good? Great. So let's do this. Everybody's good now. So we started. So ketone metabolism is in a low insulin state. You turn on this tap, HSL, fat burning and gluconeogenesis. You should know what's low insulin state in between meals, sleep, fasting, starvation, diabetes. One more common than two to have diabetic ketoacidosis. First, now please see that is ketone metabolism mitochondrial or cytosolic? Is ketone metabolism mitochondrial or cytosolic? Pathway, it's mitochondrial or cytosolic? Ketone metabolism. It is mitochondrial. That's why mature RBC cannot use ketone bodies. Liver also cannot use ketone bodies because they can synthesize ketone bodies and spare it for extrahepatic tissue. They don't have the enzyme to utilize ketone bodies. Please see the first steps up to HMG-CoA is same as which pathway? Up to HMG-CoA formation is same as which pathway? Which we'll be doing next. HMG-CoA is also formed in which pathway? Is there an HMG-CoA in pathways? Cholesterol synthesis. Please know that, except that this step is mitochondrial cholesterol synthesis, like fatty acid synthesis is cytosol. First fatty acid, there are only fatty acids and cholesterol synthesis is cytosol, ketone bodies is mitochondrial. HMG-CoA is also formed in cholesterol synthesis, difference being compartmentalization. Cholesterol synthesis is cytosolic, ketone is mitochondrial. I hope you're following my pace, everyone. Are you following my pace? Is it good?
Kind of.
Yes, ma'am.
What can I do? It's a lot of, I know I don't want, or I can go slow, but then it will take a little bit time. So you want this
No,
pace? You're good?
no, you're doing good.
Okay.
It's a material that we have to internalize, but your pace is good.
Okay, great. So know that the up to HMG-CoA formation is also in cholesterol synthesis, but cholesterol is cytosolic, ketone is mitochondrial. There are only three ketones body. First to be formed is acetoacetate. This is non-enzymatic. This is enzymatic. Acetone is expired through lungs. That's why the fruity smell in a patient with diabetic ketoacidosis. Beta hydroxybutyrate and acetoacetate in urine. Mostly beta hydroxybutyrate in urine, which is by dipstick test. But acetone, lung, fruity smell, right? When you say thioesterase, fatty acid synthesis, thiolase, fatty acid oxidation, HMG-CoA lyase, ketone metabolism. Just don't mix and mess up these enzymes because HMG-CoA reductase, same sound like HMG-CoA lyase. So pay attention on them. HMG-CoA synthase. HMG-CoA synthase can be in HMG-CoA synthase can be in, tell me. HMG-CoA synthase can be in which two pathways? Ketone synthesis is mitochondrial. Ketone metabolism is mitochondrial, not just synthesis, utilization also. The whole ketone metabolism is mitochondrial. But tell me, HMG-CoA synthase can be in not fatty acid. No, there's no HMG-CoA synthase in fatty acid. No. HMG-CoA synthase can be HMG-CoA synthase, HMG-CoA reductase, HMG-CoA lyase sound right. HMG-CoA synthase can be in ketogenesis and cholesterol synthesis. Differences compartmentalization, cholesterol synthesis cytosolic, ketogenesis mitochondrial. HMG-CoA reductase. HMG-CoA reductase, which pathway? You should have the right enzyme in the right pathway, right state, right compartmentalization, right tissue. So HMG-CoA reductase, which pathway? HMG-CoA reductase. HMG-CoA reductase, cholesterol synthesis. HMG-CoA lyase. HMG-CoA lyase. Which metabolism? HMG-CoA lyase. Ketones. Thioesterase. Thioesterase. Fatty acid. Thioesterase, fatty acid synthesis, thiolase, fatty acid oxidation. So just be mindful of the right enzyme in the right pathway, please. Acetone formation is non-enzymatic, expired keto or fruity smell. Only acidosis we are dealing with, these are these three: diabetic ketoacidosis, low insulin state, HSL is on, right? You know that. Type 1, more ketones than type 2. Diabetic ketoacidosis is more with 1 than 2. Tell me, when is propionic acid accumulating? Which enzyme deficiency or vitamin defect propionic acid is accumulating? Enzyme and vitamin, propionic acid will accumulate: carboxylase and biotin. And where will methylmalonic acid accumulate? Enzyme and vitamin. Methylmalonic acid will accumulate. Enzyme and vitamin. Methylmalonic acid. B12 and enzyme, isomerase or mutase. So please know there are different enzymes defect and different intermediates accumulating. Don't flip flop or mix them. I'm only doing for fat. We are not doing lactic acidosis. We definitely have introduced diabetic ketoacidosis, propionic and methylmalonic acid. I'm not going to talk big time about alcoholics, except I want to explain you with fat. In alcoholics, they have a high NADH to NAD ratio, so their dehydrogenases are inhibited. That accumulates fatty acids inside them and increases tags. There is a misnomer, or there is a misunderstanding among students, that tag accumulation is in the liver is because of enhanced tag synthesis. So see, this is how you got to compartmentalize. When fatty acid is formed, when there's fatty acid, for example, there is fatty acyl CoA. First of all, you should not know it as fatty acid. It should be fatty acyl CoA and glycerol 3-phosphate. Tell me, if you have fatty acyl CoA and glycerol 3-phosphate in a well-fed state, what will it form? If you have fatty acyl CoA and glycerol 3-phosphate accumulating in liver in well-fed state, what will you form? Tags. But if you are in a low insulin state, what will happen to fatty acyl CoA? What will happen to fatty acyl CoA in a low insulin state in a liver? Oxidation. What will happen to glycerol 3-phosphate in a low insulin state in the liver? Any pathway you think of? Glycerol 3-phosphate in a low insulin state. Glycerol 3-phosphate, gluconeogenesis. So you understand that each intermediate, yes, have a different fate in different state, but don't relate that alcoholics are doing tag synthesis. They are not in a well-fed state. The fatty acids accumulate in them because they are not oxidized, and glycerol 3-phosphate accumulates in them, leading to tag accumulation and fatty liver. And have you done lipoproteins? How are tags, if endogenous tag exported from liver? How are endogenous tags exported out from liver? How do you export the endogenous tag from the liver to circulation? In which lipoprotein? Endogenous tag, not dietary or exogenous. Endogenous tags are exported out as VLDL. VLDL. Alcoholics also cannot do that because they don't have protein. They are not good in nutrition. They have protein defect. They markers, and because of both of effect, tags accumulate in the alcoholics, causing fatty liver. So yes, there is low VLDL formation and there is too much tag accumulation. Others are not for this block. Also, please note, will alcoholics have ketogenesis? Will alcoholics have ketogenesis? Will alcoholics have ketogenesis? Yes. Also, please know that it's also like a low insulin state. Cholesterol. Please stick to the learning objective. Starting molecule acetyl-CoA, you know fate of cholesterol, which is the major fate of cholesterol here. Major fate of cholesterol. Major fate of membrane, please know that too. Well, how do you do cholesterol synthesis? Here also you form HMG-CoA, but in the cytosol. So which step is inhibited by statins? Formation of HMG-CoA or formation of mevalonate? Which step is inhibited by statins? Formation of HMG-CoA, formation of mevalonate. Be mindful of the steps that are physiologically significant, clinically significant. Well-fed state pathway, mevalonate formation is inhibited by statins. They are competitive inhibitor and they have multiple steps. One more thing which is relevant to statins, which we learn in pharm, is chronic use of statin causes myoglobinuria. You know why? Because one of the intermediate of statins forms CoQ, which is ETC component. So when chronic use of statins, you also reduce ETC component in your body. If the ETC components are reduced, what will go low in your body? If the ETC components are reduced, what will go low in your body? What you cannot synthesize? Yes, if you are blocking HMG-CoA reductase, you're blocking mevalonate. Very correct. Yes, if you don't form ETC component, there will be low ATP, there will be muscle cell lysis and myoglobinuria. One of the side effect of chronic statin use is myoglobinuria. You know, vitamin D is also one of the product or fate of cholesterol. Largest fate is membrane. So yes, up to HMG-CoA formation, same as ketone, except compartmentalization, cholesterol synthesis, cytosolic, ketone, mitochondrial, rate limiting enzyme for cholesterol synthesis. Rate limiting enzyme for cholesterol synthesis, HMG-CoA reductase. It is inhibited by statins. Tell me why cholesterol and bile acid also inhibit HMG-CoA reductase. Why cholesterol and bile acid also inhibit HMG-CoA reductase? Why cholesterol and bile acid also inhibit HMG-CoA reductase? Remember feedback inhibition, there's not just product inhibition, there's feedback inhibition. Are cholesterol, bile acid final product of cholesterol? Oh, sorry. Sorry. So what is the rate limiting enzyme for cholesterol synthesis? HMG-CoA reductase. Do statins inhibit HMG-CoA reductase? Are they competitive inhibitor of HMG-CoA reductase? Are statins competitive inhibitor of HMG-CoA reductase? Yes, but my question is, why does cholesterol and bile acid also inhibit HMG-CoA reductase? Feedback inhibition, because they are the final products. Remember, fate of cholesterol is to form bile acids, vitamin D, steroid hormones, membrane. So yes, the final product cholesterol and bile acid both inhibit HMG-CoA reductase. Actually, this is also used as a etiopathogenesis in gallstones. Gallstones know that bile acids and cholesterol also inhibit HMG-CoA reductase. Does that make sense? I will bring the question in the interactive session. No, SRBP very well. SREBP controls intracellular cholesterol. Please, so I will tell you what is intracellular cholesterol. So tell me, first of all, REBP is the protein that controls intracellular cholesterol. So if your cholesterol is high in the liver, what will SREBP HMG-CoA reductase? Just know one way. So if cholesterol is high in the liver, what will SREBP do to HMG-CoA reductase? What will SREBP do to HMG-CoA reductase? Activate or inhibit once it's released? What will, if you have a low, so you should know, I'll tell you what is intracellular cholesterol pool. There's a pool of cholesterol in the liver that regulates processes. So I'm asking if your intracellular cholesterol is high, it regulates it through SREBP. So when intracholesterol pool is high, SREBP will be released. And what will it do to HMG-CoA reductase? You're saying activate. No, it will inhibit it, saying that cholesterol is enough. So please know the intracellular cholesterol pool and SRBP regulation. Do one way, don't do both ways. Just do what will happen when you have high cholesterol in the liver. SREBP will be released, and what will be inhibited? What's the rate limiting enzyme for cholesterol that will be inhibited? What's the rate limiting enzyme? HMG-CoA reductase. Natural inhibitors are cholesterol bile acid, drug statins. Who controls them? SREBP. Please know HMG-CoA reductase. I have enumerated activators and inhibitors, right? Let's do lipoprotein. First, I'm just giving you quick pointers. First, know what are your tag-rich lipoprotein. Give me your tag-rich lipoprotein. Only tag-rich. Not cholesterol, not phospholipid and protein rich. What are your tag rich lipoprotein? There are only two. Kylo, give me one more. One more. VLDL, you can see that in yellow. But chylomicron is a carrier of exogenous or dietary tags. VLDL is a carrier of endogenous or dietary endogenous tags. So when they talk about tag-rich lipoprotein, don't go everywhere. It's chylomicron and VLDL only. Okay, then know their apomarkers. But who's still the carrier of maximum tags? Chylomicron. Types of tag, dietary and exogenous. Secondly, markers. What's the marker for chylomicron? Marker for chylomicron, ApoB, ApoB, ApoB48. Just remember 48. No C2E, please. Primary first, B48. When you talk about A, C, and E, they are in HTL. HTL is very kind. It's a reservoir of protein, rich in apoproteins, also give apoproteins to other lipoprotein. HDL, A, C, and E. So ACE, don't talk in chylomicrons and VLDL as the primary marker, because remember, these markers are how they will be picked up. I will also tell you what ApoC2 and E are doing, but let's first relate them to the right marker. Chylomicron, B48. What about VLDL? What will you talk in VLDL? ApoB48 or ApoB100? ApoB100. But LDL, so tag-rich chylomicrons VLDL, difference chylomicron exogenous VLDL endogenous tag, difference marker chylomicrons B48, VLDL B100. Let's go to LDL, cholesterol and cholesterol ester rich. When they talk about tag, don't talk about LDL. Only and only marker here, only and only marker in LDL. B100, very good. Apoprotein, HTL, protein and phospholipid rich. Which is the marker here? A, C, and E. Reservoir of apoproteins, very kind, gives to other apoproteins. So first delineate these lipoprotein, then bring in all the enzyme. You should know ACAT, LCAT, which is also known as PCAT, and lipoprotein lipase here. Lipoprotein lipase here. What is, so why do you need to have, the first step was in digestion, so let's not do that. The second and third digestion is with lipoprotein. So why do you want to esterify cholesterol? Tell me. Why and how do you esterify cholesterol? By adding fatty acid. So which one is polar? Free cholesterol or cholesterol ester? Left or right? Which one is polar? Free cholesterol or cholesterol ester? Which one you think is polar? Free cholesterol or cholesterol ester? Free cholesterol, because it is an OH group. So why do you want to esterify and mask that OH group? Because you want to make cholesterol inert. Where do you do that? Either to form lipoprotein or storage. Whenever you want to do storage, you will neutralize this OH group. That is why you need these enzymes. Or if you want to neutralize them and put them in lipoproteins. So when you're forming lipoprotein or when you're storing cholesterol as cholesterol, you need this enzyme to form cholesterol esters. You have too many cats working here, but this is a cat. Another cat is LCAT and PCAT. What does L and P stand for? What does L and P stand for? When I say LCAT and PCAT, L stands for lecithin, also known as phosphatidylcholine, very important phospholipid for clinically and physiologically. So these cats are esterifying cholesterol. They work in intestine or liver. In intestine to form lipoprotein and in liver to store cholesterol. LCAT and PCAT works with HTL. Also tell me, just to wake you up, if you have an LCAT and PCAT deficiency, where do you see this LCAT and PCAT? With HTL. LCAT and PCAT is mainly with HTL, extracellular enzyme working with HTL. So tell me, if you have an LCAT and PCAT deficiency, what will you see high in circulation? Just to wake you up. What will be high in circulation? If you have an LCAT PCAT deficiency, now again LDL. LCAT PCAT is not with LDL. LCAT PCAT is with HDL. Free cholesterol and also for a lot of phosphatidylcholine or lecithin, because it will not be used. Now, why are we using phosphatidylcholine, lecithin? They are just used as a source of fatty acid. So understand that this conversion just requires fatty acid. Why do you do that? To make cholesterol inert. What is required? All the cats come in here, A cat or L cat and P cat. A cat can be can be in work. A cat works in the intestine and liver intracellularly, whereas ALCAT and PCAT works extracellularly with HTL. So understand ALCAT, PCAT is with HTL. Now let's understand all the apoprotein. B48 quickly. B48 would be B48. Which lipoprotein you will connect? B48. Chylomicron, only B100. Only B100 you will talk about. LDL. VLDL also, but only B100 is LDL. Only B100 is LDL, which is very good question actually. So VLDL forms IDL and IDL forms LDL. Do you know that? How LDL is formed? VLDL, IDL, LDL. Where does this happen? VLDL forms IDL and IDL forms LDL. Where does this happen? In circulation or in the cell? VLDL, IDL, LDL conversion. It is in the circulation. Very good. That is why VLDL also has B100 like LDL, but only B100 LDL. B100 is also present in VLDL. E is to remove remnant. I'll show you what's remnants, but they also have a function. C2 activates LPL. So where do you want C2 to be? When you say LPL, LPL digests tags, right? Do lipoprotein lipase digest tag? Do lipoprotein lipase digest tags? Do the lipoprotein lipase, anything that ends with a suffix lipase, do they digest tags? Yes or no? So where do you want LPL to be? Who should have LPL? Which lipoprotein should have LPL and ApoC2? Which lipoprotein do you think should have LPL and ApoC2? Which lipoproteins have tags? No. HTL does not have tag. Chylomicrons and what else has tags? Chylomicrons and VLDL. Yes. So both chylomicrons and VLDL should require ApoC2 because ApoC2 is activator of lipoprotein lipase. And lipoprotein lipase is the enzyme that digests tags. And which lipoproteins have tags? Chylomicrons and VLDL. ApoA1 is activator of LCAT and PCAT. So where should be ApoA1? Which lipoprotein should have ApoA1? So ApoC2 should be with chylomicrons VLDL, though HDL gives them. HDL also has ApoC2. HDL is a reservoir of apoprotein, very kind. But ApoC2 is definitely required for chylomicrons and VLDL. ApoA1 activates ALCAT. What ApoA1 should be with which lipoprotein? ApoA1 should be with which lipoprotein? Tell me. ApoA1. A1 should be with which lipoprotein because it activates ALCAT. ALCAT and PCAT is with which lipoprotein? Metabolism. HDL. Absolutely correct. So ApoA1 should be with HDL. ApoC2 should be with chylomicrons and VLDL because of their function. What function these individual enzymes are doing will help you know why they should be at this lipoprotein. Remnant is for ApoE. So let's start lipoprotein metabolism. First of all, you eat tags in the diet. How are they? They are digested, absorbed. But what is the first lipoprotein to be formed which carry these dietary or exogenous tags is chylomicron. Chylomicron is the first lipoprotein to be formed. Very good to understand origin, purpose and destination. These three things, if you know lipoproteins. So let's start with chylomicron. Is hormone sensitive lipase for digestion? Please know anything that ends with suffix lipase digest tags. Is hormone sensitive lipase for digestion? Is hormone sensitive lipase for digestion? No. Digestion and absorption, is it hormone sensitive lipase? What is hormone sensitive lipase for? It's not for digestion and absorption. That's lipoprotein lipase. Yes. So what is hormone sensitive lipase for? Breakdown tags. Where? Where? Adipose tissue. So don't mix up HSL and LPL. LPL is a both LPL and what is common to them? Both digest tag. But LPL digests tags of chylomicrons and VLDL. HSL digest tags of tags. Which one is well-fed and which one is low insulin enzyme? LPL or HSL, which is well-fed? Which is well-fed enzyme? LPL. Please don't mix and mess up these two enzymes in what state they are active and where they are acting. So let's do chylomicron. You eat tags, you digest and absorb them. First lipoprotein to be formed. Never thrown into systemic circulation. First lymphatic, then systemic circulation. After a fat-rich meal, your serum becomes milky or turbid because of after fat-rich meal, your serum will become milky and turbid because of which lipoprotein? After a fat-rich meal, your serum chylomicron. Please see, you cannot see chylomicron high in fasting diabetics or in case of starvation. Chylomicrons are high after fat-rich meal. Now, who will digest chylomicron tags? Which enzyme then should punch in to clear the lipemia? Just as there is transient hyperglycemia. Transient hyperglycemia is relieved by which hormone? Transient hyperglycemia after meal is really insulin. Similarly, transient lipemia is also relieved by insulin by activating which enzyme? Transient lipemia after meals is also relieved by insulin by activating which enzyme? Tell me. What will insulin activate and relieve the transient hypertriglyceridemia or lipemia? Just as insulin relieves lipoprotein lipase. A lipoprotein lipase digests tags of chylomicrons and VLDL. But chylomicrons are high right after meals. That's why just after meal, your serum can be milky or turbid because of chylomicron. Just as transient lipemia is relieved by insulin, transient hyperglycemia is relieved by insulin. Transient lipemia is also relieved by insulin by activating lipoprotein lipase. There is no HPL. This is it. The other enzyme is HSL, please. There is LPL and there's HSL. Yes. HSL is different, absolutely. So chylomicron is the first lipoprotein to be formed, released after a meal. Your serum will be turbid, but the lipemia will be relieved by insulin by lipoprotein lipase. So origin, destination, and let's do that for every lipoprotein. So chylomicrons are formed in the intestine, yes. What do they carry? Endogenous tag or exogenous and dietary tags? What do chylomicrons carry? Endogenous tag or exogenous or dietary tags? What tags chylomicrons carry? Endogenous tags or exogenous or dietary tags? Exogenous and dietary tags. Where do they go? Destination, adipose and muscle. What is hanging in there in the capillaries of adipose and muscle? Lipoprotein lipase. Look at this here. HDL is very kind, is giving us a C2 and E. Why? What will C2 activate? What will C2 activate? Why is HDL giving C2? Why is HDL giving C2? C2 will activate LPL. Very good. And what will ApoE do? It will recognize remnant to be picked up. So now when all chylomicrons are digested, tags are digested by LPL. What is this known as? After all tags is digested by lipoprotein lipase, chylomicron now known as, after LPL has acted on it, chylomicron is now known as, when all the tags is digested, now chylomicron after LPL digestion is known as chylomicron remnant. What is its fate? It's picked up by the liver by ApoE. So ApoE is for remnants. Do we have VLDL remnant too? Yes. So this finishes chylomicron. Origin, intestine. Marker, B48. Carrier, dietary tags. HDL, very kind, gives apoprotein. Why do you need ApoC2? LPL. Why do you need ApoE? Remnant. What is their destination? Adipose and muscle. What is hanging in there? Extracellular enzyme, lipoprotein lipase. Digest tags, resultant product, remnant. Remnant picked by liver, ApoE receptors. This is chylomicron. But now what is VLDL? VLDL is not carrying exogenous or dietary tags. VLDL, look at, is carrying the endogenous tags. So tell me, where is tag synthesis? Major, DNL. Where is the major tag synthesis? That is endogenous tag. That's not dietary tags. Where is the major tag synthesis? DNL. Liver, but they can be kept there, otherwise there'll be fatty liver. We all would suffer from fatty livers. So these tags need to be exported out. Who does that? VLDL. Alcoholics cannot do that. That's why they suffer from fatty liver. Here also, please differentiate tag synthesis between liver and adipose tissue. I have addressed that in my last section. You must understand tag synthesis between liver and adipose tissue. But right now we are talking about major tag synthesis, which is liver, and how is to be exported out. Where is major tag synthesis? Liver, exported out as VLDL. It requires the same apoproteins. So which is the apoprotein in VLDL? Is it mainly B48 or B100? VLDL. VLDL is B48 or B100? VLDL is B48 or B100? VLDL. VLDL is B48 or B100? B100. Does it need C2? Yes. Just as lipoprotein lipase digests chylomicron tags, they also digest VLDL tags. Different. Chylomicron is after meals, postprandial, and VLDL is post-absorptive, right? They have the same fate. Lipoprotein lipase digests tags. Now, please be mindful. Chylomicron after lipoprotein lipase acted, what was the product? Chylomicron after LPL acted, what was the product? After LPL acted, what does chylomicron change into? Once LPL acts, chylomicron changes into chylomicron remnant. But you know what is VLDL remnant? IDL. IDL has two fate, which is not shown in this slide. IDL can be picked up by liver. What are remnants picked up by? What apomarker? IDL has two fate, which is not shown in the slide. ApoE, absolutely right. But there is one more fate of IDL. It forms LDL. What is the only marker in LDL? B100. Does LDL also have two fate? Absolutely. Please know this fate very well, which is picking up by B100 receptors. Are B100 receptors highly regulated? Are B100 receptors highly regulated? Are B100 receptors highly regulated, which is for LDL? Yes, please know the difference between highly regulated B100 receptor and scavenger receptors. I will tell you coming up in the slide. You must know B100 are highly regulated. But there is one more fate of LDL, to give cholesterol to peripheral tissue. Now, this can be bad. Otherwise, it's normal, but that's why it's known as bad cholesterol when you're missing this fate. If LDL is not picked up by liver, what will happen? Tell me. If LDL is not picked up by liver, what will happen? All the cholesterol will go where? If LDL is not picked up by liver, peripheral tissue, extrahepatic tissue, peripheral tissue, is it good? No. This can be atherogenic. Where is this seen in? Familial hypercholesterolemia. Familial hypercholesterolemia, LDL receptor copies are low. So this fate is missing. So LDL is not a bad cholesterol. It becomes bad pathology when this fate is missing. All the cholesterol will go to the peripheral tissue. Now comes scavenger receptor. You must know that to pick up LDL, first of all, who is atherogenic? LDL or oxidized LDL? Is LDL atherogenic or oxidized LDL? Is LDL atherogenic or oxidized LDL? Who is atherogenic, LDL or oxidized LDL? LDL, oxidized LDL. And to pick up oxidized LDL here in the peripheral tissue, receptors are known as scavenger receptors. Scavenger receptors are not regulated. That's why they are dangerous. They will pick up everything that is given to them. They are not regulated. That is why delivering more and more LDL to the peripheral tissue is dangerous. That's why it's known as bad cholesterol. It delivers cholesterol towards or away from peripheral tissue. LDL is delivering cholesterol towards or away from peripheral tissue. LDL is delivering cholesterol towards or away from peripheral tissue. Towards or away? Where is LDL delivering cholesterol? Towards peripheral tissue or away from it? Yes? not away, towards peripheral tissue. That's why it's a bad cholesterol if this fate is increased. HDL delivers cholesterol away from peripheral tissue. So let's see HDL. HDL is good. Look at HDL. HDL is moving all the cholesterol from peripheral tissue back to the liver. But HDL is reservoir of apoprotein. Who activates ALCAT or PCAT? Who activates ALCAT and PCAT? ApoC2 was for lipoprotein lipase, and ApoA1 is for ALCAT and PCAT. Remember the ALCAT and PCAT, what do? They convert cholesterol into cholesterol ester, make them inert, and move them more and more inside. So you need PCAT, you need ApoA for HDL metabolism. But HDL is a good cholesterol. It moves cholesterol away from cholesterol, peripheral tissue. What is that known as? Moving cholesterol away from peripheral tissue is known as reverse cholesterol transport. So understand that LDL moves the cholesterol towards bad cholesterol, towards the peripheral tissue. HDL moves it away from peripheral tissue. Also know oxidized LDL receptors are scavenger, right? So now you know the intracellular cholesterol pool. Let's do SRE, BP, and statins. Very important here, please. Let's see first what is the contributor to cholesterol pool. Does LDL bring cholesterol to the liver? Does LDL bring cholesterol to the liver? Does LDL bring cholesterol to the liver? Is one of the fate of LDL to bring cholesterol to the liver? Yes. Does HDL bring cholesterol to the liver? Yes, it moves cholesterol away from peripheral tissue to the liver. Very good. Do diet when you eat. Now you will see here that it's not that that when you say that there is a familial hypercholesteremia, it runs in the family. It's also because no one runs in the family, right? It's not that everything runs in the family. It's also sometimes no one runs in the family. You're not doing exercise. You're not doing proper diet. I'll show you why. So the major contributor of cholesterol are HDL and LDL, but whatever you eat in diet is also contributing to cholesterol. Now, why is this dangerous? You should understand intracellular cholesterol pool, SRE, BP, and statin. For example, if your intracellular cholesterol pool is high, what will be down-regulated? Tell me. Just do one thing. If your intracellular cholesterol pool will be high, what will be down-regulated but through SREBP? First of all, HMG-CoA reductase and also LDL receptors. If your intracellular cholesterol pool is high, this is normal. Now let's do statins. So what are statins doing? Are they inhibiting HMG-CoA reductase? What are statins doing? So first, normal regulation. When intracellular cholesterol pool is enough, two things are down-regulated through SREBP, HMG-CoA reductase and LDL receptors. Now, let's talk about statins. If statins inhibit HMG-CoA reductase, what will happen to LDL receptor? If statins inhibit HMG-CoA reductase, what will happen to LDL receptor? Upregulated or downregulated? If statins are inhibiting down-regulated or upregulated, upregulated. Again, first know normal to know abnormal. So what is intracellular cholesterol pool? When it is high, then you don't want LDL to bring more cholesterol. You don't want de novo cholesterol synthesis. So when intracellular cholesterol pool is high, so let's see what all is contributing to this pool. What all is contributing to this pool? LDL, HDL, whatever we eat in the diet, or also your de novo synthesis. So yes, when your intracellular cholesterol pool is high through SREBP, two things will be down-regulated, HMG-CoA reductase and LDL receptor on the surface. But now let's look at statin. If statin inhibits HMG-CoA reductase, what will happen to LDL receptor on the surface? What will happen to LDL? Increase. Is it beneficial? Is it beneficial to? Yes, because you will bring more an LDL inside the liver for utilization. Please understand this function is very important. The B100 are highly regulated. It's important to bring in LDL inside the liver and increase the utilization of the cholesterol that it is carrying. Because if this does not happen, there will be too much LDL in the circulation, too much cholesterol, and that is atherogenic. These LDL receptors have to be healthy, be in good copy, functional to prevent atherosclerosis. You know why? If LDL is not picked up by liver, where it is going? Why are you worried about that? If LDL is not picked up by liver, where is it going? Which is the other fate of LDL? Peripheral tissue, and it will be atherogenic. So understand intracellular cholesterol pool, its regulation through SREBP, understand drugs because it is highly regulated by B100 receptors. Now, what about HDL receptor like oxidized LDL? They are scavengers, they are not regulated. So two receptors are not regulated. The receptors in peripheral tissue for oxidized LDL and the receptors on the tissue for HDL. Please know that very well. They are not regulated. Scavenger receptors are two places for oxidized LDL and HDL for uptake by tissue. Only way you can get rid of cholesterol is bile, too much cholesterol to bile, gallstone. So yes, LDL receptor have to be functional and healthy to move LDL inside the liver for utilization. This is very important so you don't accumulate LDL in the circulation. More and more LDL in the circulation, more chances of atherosclerosis. Also, this is also important because students not see Niemann type A and B are important, which I will address in phospholipid, but students are confused with Niemann-Pick type C. It's not the same as A and B. C is a defect in exporting cholesterol into the liver and contribute to the intracellular cholesterol pool. That's the C defect. Please don't confuse Niemann-Pick type C with A and B. Please, they are different. Right? So do the SREBP, intracellular cholesterol pool very well, understand their regulation, do only one way. If you have too much cholesterol inside the cell, what will be downregulated? If you have too much cholesterol inside the cell, what will be downregulated? Two things will be downregulated. If you have too much cholesterol inside the cell, what will be downregulated? Number one, HMG-CoA reductase. Number two, what else will be downregulated? LDL receptors on the surface. Just know one way. I have drawn it in the diagram. Either know right side or left side. Don't do both ways, please. Let's do this one. Let's review this. These are my last slides. Please pay attention on that. This is the whole summary of lipoprotein. If we will, I will open question and answer for Monday. Do this in your weekend. Now let's look at this. Let's look at the normal function. Our endogenous tag synthesized in the liver, yes, is major site for de novo lipogenesis liver. Yes, that will go into tag synthesis. Who will export these tags outside the liver? Which lipoprotein? Which lipoprotein exports exogenous tag from liver? VLDL. What's their destination? They have to go to peripheral tissue, which one? Adipose and muscle. But before that, they have to be metabolized. What does lipoprotein lipase do to VLDL? So VLDL endogenous tag is exported out by VLDL. What's hanging in the blood vessel of adipose and muscle? Lipoprotein lipase. Will they digest tags in VLDL? Will lipoprotein lipase digest tags in the VLDL? Yes. What will they form? What is VLDL remnants known as? So lipoprotein lipase will digest the VLDL tags and form what? IDL. Does IDL have two fate? This slide is showing that fate. This is all normal. Either it's picked up by liver or converted into LDL. So please see that VLDL, IDL, LDL conversion is in the circulation, not in the tissue. Does LDL have two fate? What are these receptors? 300 regulated receptor. Does LDL have two fate? Does LDL have two fate? What are these receptors here, which are where they are picked up by liver? It is scavenger or regulated receptors on the liver. Regulated receptor. What's the other fate of LDL? Delivers cholesterol to the peripheral tissue. That's why LDL is a bad cholesterol, not normally, only when this fate is not happening. If this fate is low, LDL can be atherogenic, right? ApoB100 are highly regulated. LDL is a bad cholesterol, not because it's delivering cholesterol towards the peripheral tissue, because this fate can be bad if A is missing. It's like enzyme deficiency. When normal pathway is missing, alternate pathway will turn on. So now let's see LDL receptor defect. If you have an LDL receptor defect, what disorder have you learned? What disorder have you learned where there is low copies of LDL? Any disorder or disease you have learned where there is low copy of LDL? Familial hypercholesterolemia. Yes. Will LDL be taken up by the liver in familial hypercholesteremia? No. What will happen to LDL? Increase in circulation. More and more chances of LDL to become oxidized LDL and taken up by scavenger receptors in the peripheral tissue. This is atherogenic. Please know the difference between B100 receptors and scavenger receptors. So yes, if you have low number of copies of LDL, there will be more and more LDL in the circulation, more chances of oxidized LDL, more atherosclerosis. Please know that it's not just it runs in the family. If no one runs in the family, it's the same. If you eat too much cholesterol in the diet, it will also mimic familial hypercholesterolemia, not into the same extent, but yes, to some extent, because high cholesterol consumption in the diet also increases intracellular cholesterol pool, down regulating LDL receptors, which will also be atherogenic. So it's not that it runs in the family, it's also because no one runs in the family. Exercise and diet is also important. They mimic same like familial hypercholesterolemia. Familial hypercholesterolemia, LDL receptor defect, which is which apoprotein? Familial hypercholesterolemia, LDL receptor defect, which is which apoprotein, please? ApoB? ApoB? LDL receptor defect, ApoB100. And there are low copies of ApoB100. What was high in the circulation? LDL. What is LDL carrying? Cholesterol. That's why it looks orange colored. If in familial hypercholesterolemia cases, cholesterol gives an orange color. You can also separate it from electrophoresis. Familial hypercholesteremia, which lipoprotein would be high? Chylomicrons, VLDL, HDL, or LDL. Familial hypercholesterolemia, which lipoprotein will be high? Chylomicrons, VLDL, LDL, or HDL. Look at this beta band. This is also known as LDL. You can see it by electrophoresis or measuring it. So familial hypercholesteremia, homozygous form, especially, I want to put that note. So familial hypercholesterolemia, what lipoprotein is high? Familial hypercholesteremia, which lipoprotein is high? Chylomicrons, VLDL, LDL, HDL. Familial hypercholesterolemia, which lipoprotein is high? Which lipoprotein is high in familial hypercholesteremia? LDL. Which marker is defective? ApoB100. What is high? Cholesterol, not tags here. That's why there are drugs that will, all drugs that you will give to familial hypercholesteremia is to lower cholesterol. But there is one more protein which is very important apart from LDL and VLDL, which is PCSK9. Now, please, just to wake you up, PCSK, no normal to no abnormal. PCSK9 degrades LDL receptors. So tell me, gain of function of mutation of PCSK9 or loss of function of mutation of PCSK9 hypercholesteremia. Again, no normal to no abnormal. PCSK9 degrades LDL receptors. So gain of function of mutation of PCSK9 or loss of function of mutation of PCSK9 will lead to familial hypercholesterolemia. Very good, excellent. Gain of function of PCSK9 leads to familial hypercholesterolemia. That is why PCSK9 inhibitors is used as a treatment. You can give any hypocholesterolic drugs, please, but for homozygous familial hypercholesterolemia, PCSK9 inhibitors work better. Know that niacin is which vitamin? Niacin is which vitamin? B1, B2, B3. Niacin is which one? B3. It's also hypocholesteremic drug, just for your information. So know the function of PCSK9, and that is why anti-PCSK9 therapy is helpful in familial hypercholesterolemia. Lastly, ApoE2, E3, or E4. Which one is for lipoprotein metabolism? E2, E3, or E4? Which one is which ApoE is for the ApoE of lipoprotein is E2, E3, or E4? Tell me. E2, epsilon 2, epsilon 3, or epsilon 4? Which one is for lipoprotein? 3. 2 and 4 are for Alzheimer's. 3 is the ApoE marker. That's all for today, and I wish you good luck.
Thank you, Dr. Bach.
You're welcome.
So
Dr. Bach,
I have-
was this recorded? in
Yes,
case
tell me,
we want to
tell
watch it
me.
later?
Yeah, tell me, Diego.
No, you go ahead, Miss Abrams.
Oh, I
Yes,
just
tell-
want to know, was this recorded, like in case I want to watch it later or no?
This was not recorded because I did not have any request for recording, please.
Oh, okay. Well, thank you so much for today.
No, but I shared this slide, and also please note I am opening the question answer session on Sunday. Why I am doing this weekend? Because it's these 4 lectures, I want you to get them off your plate. So I'm going to open Sunday question and answer for these 4 lectures, but I'm going to redo it in the interactive, so don't worry.
Okay,
I'll
do
come
you know
back
what
with these
time have
slides.
you decided for the Sunday one?
Sorry?
What time did you decide for the Sunday?
I don't know whatever students want. I just want to
Oh.
open question and answer. I think 10 is good. What do you think? 10 Sunday?
Sounds
If you have any
reasonable.
question, sorry.
Sounds reasonable.
Okay, so 10 Sunday using the same link. Let these 4 lectures off your plate. Read and come and ask for question. There will be no review, please. This is only for question. Okay? Great. Sunday 10. See you Sunday 10. This Sunday, not next. Just
One question. So right now our schedule still shows something going on Monday from 3 to 5.
Yes, that is your
Okay.
interactive session. Yeah.
Okay.
Your
Um,
I have not done your interactive, right, Diego? Your interactive sessions are 2, if I'm not wrong. One is on Monday, starting Monday.
got it. Another question regarding the slides, the lecture slides versus this compilation that you gave us. Should we maybe focus more on these slides to get a grasp of the content, or
Yes, because
perhaps
yes, because I have explained you through this different concepts. Take them as a focus, but I would recommend your learning objective as the key focus. But take them as a focus. I think I have explained them well. So this will help you with the focus. Yes. These recap slides. Right?
Mhm.
What else? Anybody has question? Everybody's good? Okay.
Thank you.
Okay, great. I'll share. I think you have these slides now, right, everyone?
Yeah, we do.
Great. See you Sunday, 10.
Thank you very much. Have a good day.
Thank
You
you.
too. Bye.
Have a good day.
Okay, bye. I think Vishal and Lauren. Are you good? Vishal, Maya? They're still around?